Hi everyone, this is our third and last lecture on chi-square tests. We'll talk about tests for homogeneity. This is the third type of chi-square test to go along with tests for independence and goodness of fit tests. Let's talk a little bit about tests for independence versus tests for homogeneity. How are these tests different? First off, mathematically, they are exactly the same. And as far as what you do in StatCrunch, they are also going to be exactly the same. For both, you will go to Stat, Tables, Contingency, and when you see the pop-up box, you will click chi-square test for independence. There's not going to be a separate line to check for tests for homogeneity. Since they're mathematically the same, StatCrunch just has you check the independence choice for producing the results. Okay, so what are the differences? A test for homogeneity is set up like a two-sample proportion test. It's going to involve two populations and two samples that you will collect, one from each population. Okay, so one of the features of a test for homogeneity will be that your contingency table is going to have two columns, one column for each sample. The word homogeneity just means similar or like, and really what you're looking for is, are these two populations similar or are they not similar? And that's similar to what we did in a two-sample proportion test. In a two-sample proportion test, we collected two samples from two different populations, and we compared the proportions between those populations to see if they were close enough to be considered the same. This is going to be similar to that. It's just that instead of a single proportion, we might have a number of factors from the other categorical variable that you're looking at. Okay. Another difference between tests for independence and homogeneity are the hypotheses. For a test for homogeneity, again, this will be in words, the null hypothesis will be the distributions of the two populations are the same. The alternative hypothesis will just put in the word not. All of your hypotheses for tests for homogeneity will look like this. Okay. You might specify what the two populations are, male versus female, Republican versus Democrat, whatever it might be, but you will word the hypotheses this way. All right, let's look at an example. Let's look at Republicans and Democrats. Do Republicans and Democrats feel similarly or not about global climate change? Okay, so our two populations are going to be Republicans and Democrats. Okay. Here is some data produced by Pew Research. This was a survey. It was actually done back in 2018. You can read that at the bottom of the graphic box there. Okay, and there's actually kind of two questions here. They're looking at the percent of U.S. adults who say policies aimed at reducing the effects of climate change generally do more good than harm to the environment. Do policies do more harm than good? And do they make no difference? Okay. And then there's a second question under that about whether policies are good, helpful, or hurtful to the economy. Okay. We'll look at the first of these two questions. All right, now, as in the example we had on the COVID virus and blood types in China, they're giving us the information in percentages. So if we know the sample sizes, how many Republicans and how many Democrats took part in the survey, we can convert these percentages to frequencies, and we'll need frequencies in order to run the test. And as I did this, I had to do a little bit of estimating. You might notice the percentages in these bar graphs don't necessarily add up to 100%. There was a little rounding involved in creating the percents, so I had to do a little bit of adjusting to create the frequencies. The sample sizes, there were exactly 981 Republicans or leaning Republican and 1,483 Democrats or leaning Democrats. All right, so here is the table of frequencies that I created from those sample sizes and the percentages. All right, now we can run the hypothesis test. All right, so here the general question is, do Democrats and Republicans feel similarly about whether government policies do more harm or good for the environment? Looking at the percentages on the previous slide and the results here, it looks like they probably do not feel similarly. Let's see what we get from the test. Our null hypothesis is the distributions of Republicans and Democrats are the same that you see in those columns. The alternative hypothesis is that the distributions are not the same. Let's go ahead and use a level of significance of 0.05. This will be a chi-square distribution with 2 degrees of freedom. We've got 3 columns, 2 columns and 3 rows. 3 minus 1 is 2, 2 minus 1 is 1, 2 times 1 is 2. There is your degrees of freedom. Okay. All right, from StatCrunch, you would put this information into StatCrunch as a table and have it crunch the numbers, and we get a very extremely large chi-squared statistic, 352. The p-value is reported at less than 0.001, essentially 0 close to 0. So our decision is to reject the null hypothesis. And our conclusion is we have sufficient evidence to conclude that the distributions of Republicans and Democrats with respect to this question are different. There is a difference among Americans on climate change policies, how they look at those, at least on this question with respect to party. Okay, so that's a test for homogeneity. Again, it looks a lot like test for independence. Mathematically, and what you do in StatCrunch is going to be exactly the same. You will state the hypotheses differently and the conclusions differently as well. All right, now one additional topic here. So far, in all the chi-square tests we've done, we've picked summary. Okay, you can do chi-square tests with raw data, and the example here I have is survey data from a previous Math 200 class. Okay, each row represents a student, and there's 3 questions represented here. We asked students what their favorite color was, what year they were in school. We gave this as a number, but the numbers represent classes: senior, junior, sophomore, freshman, and then what major they had. When you go in to StatCrunch, you go to Stat, Tables, Contingency, and just pick with data instead of with summary. So you can do this for either a test for independence or a test for homogeneity. Okay? Just pick any 2 of the columns. When you click and you get the pop-up box, you have 2 choices to make. You need to pick a row variable and a column variable. Just any 2 of the categorical variables that you want to test. Okay? We're going to look at favorite color versus major. We're basically going to check, is a student's major independent of their favorite color? We would probably think yes, color and major shouldn't have anything to do with each other. We wouldn't expect blue-eyed people to decide to be athletic trainers more than green-eyed people. If you were doing a test for homogeneity, you would need to pick your column variable to be a variable that only had two values, male and female, for example, Republican or Democrat, for example. Okay, but you could do that. We can do tests for homogeneity. All right, and when you click that, you get results just like before. At the bottom, we can see it's a chi-square test. The degrees of freedom is 9, gives you chi-square value of 5.2 and a p-value of 0.815. That's a large p-value. It would certainly not be less than 0.05, so we would fail to reject the null hypothesis. It looks like favorite color and major are independent. They don't have any variation or dependence on each other. Okay? And as a bonus, StatCrunch creates the contingency table, what we've been using as summary tables. Okay? So the integer numbers there, next to blue we've got 5 for business, 15 for nurses, 8 for other. Those are the counts that you would get summarizing the data that appeared in those columns. And I also checked at expected counts, those in parentheses underneath. All right, now one warning at the bottom, and this applies to all chi-square tests, independence, goodness of fit, and tests for homogeneity. Okay. There's a warning at the bottom that says over 20% of the cells have an expected count less than 5. Generally speaking, ideally you would like all of the expected counts in your chi-square table to be at least 5. Now that can be a problem if you have small sample sizes, and in this case we do. StatCrunch uses 20% of the cells being below, having counts below 5 as sort of a warning. Some authors would say they should all be at least 5. Okay, so in a case like this where you don't meet that criterion, you would simply report that fact as part of your conclusion. Okay, so you might say, our test indicates that favorite color and major seem to be independent of each other, but our expected counts are not all less than 5, so we have less confidence in our conclusion than we otherwise would. And that's how you might state that. All right, so that's the last of our 3 lectures. This should be enough for you to complete all the problems in the homework assignment. Again, I recommend you looking at the book as you need to, but these 3 lectures together should tell you pretty much all you need to know for this chapter. Please feel free to email or do a Teams call if you have questions as you are working through the homework problems. We'll see you soon.